brain-body interaction seminar series, build a really nice community of people who are interested in how the nervous system interacts with different organ systems within the body. And we've looked at that from all kinds of different angles over the year. And next year we'll continue, starting in January, with many different interesting angles on how the brain interacts with the body. But this week, we are going somewhere that we haven't covered yet. So, we've looked at how the brain interacts with the immune system and with the gut and the microbiome, and amongst others, but we haven't yet explored how the brain interacts with the skeletal system. And this week we're going to rectify that. So we're very honored to be joined by Professor Gerard Karsenty. So Professor Karsenty is the Paul A. Marks Professor and Chair of the Department of Genetics and Development at Columbia University in New York City. So just to give a brief introduction, I think Professor Karsenty's work has really led the field of bone physiology and development. And so he did his undergraduate and MD PhDs in France before moving to start his own lab in the USA, first at the University of Texas and then at Baylor College of Medicine in Houston. And then in 2006, he took up his current position at Columbia University. And during his time there at Columbia, his lab has studied various aspects of the biology of the skeleton from all kinds of different angles. So his lab has a large body of work looking at the genetics of how osteoblasts differentiate and how bone develops. And also how the mass of bone is coordinated with energy balance and reproduction. And of relevance to our series, his work more recently has looked at how bone interacts with all kinds of different organ systems in the body, particularly with the brain. So, for example, he's shown that bone physiology is regulated by specific circuits in the brain, and he's explored the idea of bone as an endocrine organ in its own right, particularly via the secretion of the hormone osteocalcin. And he's shown roles for that hormone in brain development, in the acute stress response, in male fertility, in glucose metabolism, and more. And I'm sure we'll hear a lot about that today. So without further ado, I'll hand over to Professor Karsenty. Just if you have a question, feel free to write it in the Ask a Question box at the bottom. And if you see another question that you like, you can vote for that question. And then at the end, we'll go to a question and answer session.
Thank you, Sam, and also thank you to Maria and to this organization for having invited me. I think it is a very original and intellectually powerful organization, and you have to be congratulated to have put that together. So I wanted to start with the logo of your group, but it does not come on the screen. That does not really matter. And I wanted to start with it, not so much, in fact, not at all because bone is not on the logo, but because it puts brain at the center, at a preeminent organ, and all the other organs as not slave, but kind of slave of the brain. And I would like to correct that if I can today. And I start this talk by this citation, which to me is absolutely extraordinary because it was written in 1969 by a scientist who had no medical training and who was working in bacteria all his life. And what he said is the best definition of physiology that I know. Any organism is a functional unit, coherent and integrated. And humbly, we are far to have in complex organisms he knew nothing about, elucidated the entire structure of these systems. So this was written, again, by a bacterial geneticist in a book that is unreadable now, but to me it is a strikingly intelligent conception of what molecular biology should lead to. And this is why I make this point, is that maybe the brain is in the middle, or maybe there is no middle, and everybody is as important than every organ is as important than any other organ because of this integration between organs that occurs in complex organisms. So during my talk, I will try to explain as much as I can why is it that we do what we do, and then to go over several aspects that have brought us again and again and again to link the skeleton and mostly the bone to the brain in various directions. So to start with, I would like to emphasize the dichotomy and the contradiction that there is in studying organ biology. When people like me who are studying one organ all their life essentially ask always, in every field, the same question. These questions can be asked in an infinite number of ways, but they are fundamentally the same question. The first question is, how does cell differentiation occur and leads to the formation of this organ? How do we go from an inner mass to a liver? And this is the topic of developmental biology. And the second question is, how does this organ fulfill its function? And you could say it is the domain of physiology. These two questions are fundamentally opposed in what they address. In the question of developmental biology, you could rephrase it by telling, we don't know how cell differentiation and organogenesis occur, and we want to know how it occurs. Whereas how I phrase the question of physiology, you could say, we know exactly what the liver does, and all we want to do is to have the molecular basis of this function. And there is a profound opposition between these two questions. And then there is a third question, which is the one we address, which may sound wild, but turns out that it is in fact the most fertile one in physiology, which is, do we really know all the function that the organ we study fulfill? And the answer, as you will see, at least for the skeleton, and the skeleton is no exception, the answer is no. So when it comes to the skeleton, the question can be rephrased, and it says, does bone have any other function besides making bone? Behind this question, there is another one looming, which obviously we did not have in mind when we started, which is, why exactly did evolution invent bone when animals left the sea to go on the land? And there are many answers to this question, but some of them are certainly unexpected. And so the reason we ask, why does bone have any other function besides making bone, is a particular feature of bone, which is that it is the only organ that contains a cell type whose only function is to destroy the host tissue. Of course, we have lymphocytes everywhere, macrophages, but most of the days they don't destroy our liver or other organs, whereas the osteoclast was invented by evolution precisely to destroy bone. And it occurs in a binary physiological function that is called bone modeling during childhood and bone remodeling after childhood. So in this function, there is destruction is always followed by formation. And this is a process whereby bone renews itself throughout life. So it allows growth, and an animal that grows can walk. In other words, an animal that cannot grow cannot walk. And an animal that cannot walk, even in the 21st century, pandemic or no pandemic, cannot live. So bone modeling is a survival function. It fulfills all the definition of a survival function. During adulthood, bone remodeling now repairs micro and macro damage, and essentially was the only orthopedic surgeon that was living on this planet until the beginning of the 20th century. So those are not trivial functions, and you can imagine the cost in human life of any war in the world if there was no bone remodeling in the 19th or 18th or even early 20th century. It takes a lot of energy to commit suicide, and it takes even more energy if after committing suicide, you want to be reborn. But whatever is this amount of energy, it is proportional to the surface covered by the organ. And bone covers more surface than the spleen, the retina, or even the lung. So the energetic cost should be very high. And it can be verified not necessarily or only using complicated and in fact difficult, given the nature of the tissue, experimental technique, but with another experimentalist, probably the most reliable one, which is medicine. Any condition restricting food intake causes a growth arrest in children. And in adults, it causes osteoporosis. At the same time, we know that gonadal failure causes bone loss in women, but also in men. So based on this unusual reading of bone physiology and on this clinical/experimental observation, we proposed 20 years ago this hypothesis that there should be a coordinated regulation, endocrine in nature because the organs are not next to each other, of bone growth, energy metabolism, and reproduction. And I would like to insist here on what this hypothesis that seems pretty vague is in fact very restrictive and far-reaching. If this hypothesis is right, these hormones, whatever they are, have to appear during evolution with bone, because it's the very existence of bone that justifies their function, or if not their existence. And again, if this hypothesis is right, bone cannot be only a sponge, but it has to be an endocrine organ that would regulate energy metabolism and reproduction. This is not the first aspect of the hypothesis that we worked on. It's a second one, but I will come to it in a minute. In the first one, what we showed is that adipocytes, which appears during evolution with bone, make an hormone which appears during evolution with bone. There is no leptin in functional terms in any animal that has no bone, that act in the brain in the brainstem and inhibit serotonin synthesis, and as a result, through pathways we identify, inhibit melanocortin and appetite, and inhibit the sympathetic tone and bone mass accrual. And I boxed in blue the sympathetic tone and bone mass accrual, not because bone is more important than appetite. There is nothing more important than anything else in physiology. But because based on this work, there is now 18 years after we established this paradigm, an NIH-based nationwide clinical trial to demonstrate that beta blocker could be used not to treat osteoporosis, but to eradicate osteoporosis altogether. And that's how mouse genetics can affect human medicine, hopefully. So that's what we showed. And indeed, there is a fat-derived signal that co-regulates bone mass and energy metabolism. But as I said, the most provocative aspect of this hypothesis is that bone should be an endocrine organ. And that's what we tried to show following the work on leptin and bone. And what we showed is that there is an hormone called osteocalcin that is an osteoblast-specific gene. And it's important because all, if not most, if not all, peptide hormones are cell-specific genes that encodes a secreted protein that has every structural feature of a hormone. The business end of osteocalcin is made of 3 glutamate residues that, when they are carboxylated, make the protein inactive as an hormone. And we were fortunate when we studied osteocalcin to rely on 2 mouse models that were a control for each other. A loss of function model, the osteocalcin knockout, and a gain of function model that is called by another acronym, ESP null mice. And this hormone is osteocalcin. And again, I'm going to skip what is not related to the nervous system, but initially we showed that osteocalcin acts on beta cells to favor insulin secretion after it binds to its receptor GPRC6A, and acts on Leydig cells to favor testosterone synthesis and male fertility. And again, the receptor is the same. The red diamond here is here to indicate that mutation in GPRC6A in humans are associated with impotence in humans and glucose intolerance. And so the hypothesis, you could say, was verified fully. We had the hormone, we had the receptor, we had the mouse data and the human data. And we had more. What we had is that energy metabolism acts in the brain and the brain regulates bone mass, which raises a question. Does bone, in a feedback loop, speaks to the brain? And if it does, what does it do? And because the only tool we had, and we still have, is osteocalcin, we ask this question in terms of osteocalcin biology. Does osteocalcin signal in the brain? And if it does, for what purpose? And the first experiment we did to address this question was to determine if osteocalcin crosses the blood-brain barrier. So what we did was to take osteocalcin null mice, so there is no osteocalcin around, and implanting them pumps in their back, pumps that deliver osteocalcin 300 nanogram per hour. And then we ask, what happened in the brain? Can we find osteocalcin in the brain? And you can see on the right that, of course, osteocalcin was found in the serum and in the bone because it has a tropism for bone. But it was also found in the midbrain, the brainstem, and the hypothalamus. indicating that osteocalcin can cross the blood-brain barrier and accumulate in some part of the brain. Osteocalcin is neuroactive. If you drop osteocalcin on neurons in vitro, you get a response in terms of calcium sensing and depolarization of the neurons. And osteocalcin has a profound effect on the anatomy of the brain. In the absence of osteocalcin, the corpus callosum is gone, and the structure of the hippocampus is quite different and much smaller than it is in wild-type mice. And we can, in fact, measure the area of the gyrus dentate in knockout of osteocalcin, and it is decreased, approximately 30%. Molecularly, we see that in the absence of osteocalcin, the content of serotonin in the brainstem and in midbrain is decreased approximately 40% in the brainstem. The content of serotonin of norepinephrine is decreased. And more strikingly, the content of dopamine is decreased close to 70% in midbrain and striatum. So what does it do to the mice? So we measured anxiety in these mice using a classical test, and I don't really need to speak. You can see that the osteocalcin mice have an obvious phenotype of anxiety. We also measure spatial learning and memory, and you will appreciate why these mice, for those of you who went through medical school, why these mice cannot even consider going to medical school. So in this Maurice water maze, as you know better than me, mice have to find a platform that is under milky water. Mice are virtually blind and they don't like water. And over a course of a 4 to 5 days period, as you can see in wild-type mice on the left, they learn how to find the platform pretty quickly. I could leave the slide for 15 minutes. The osteocalcin mice will never find the platform. So that's the intensity of the impact that osteocalcin has on not only anxiety and depression that I did not show, but memory. And this is another quantification of this assay, where you can see that wild-type mice within 3 days learn where is the platform. I'm sorry. Whereas the osteocalcin null mice for 12 days learn nothing. And remarkably, osteocalcin heterozygous mice also have a phenotype, which is very rare for an hormone. So nominally, this tells you what osteocalcin does to the brain. But in essence, it tells you something much more. It tells you that what bone is doing to the brain, and that in the absence of bone, well, there is no memory, and there is an increase in anxiety. And this refers to what I was telling you at the beginning. It goes both ways. Bone is a slave of the brain to measure bone mass, but brain, in all due respect, depends on bone for its functions. So I'm not going to show all the data, but I want to show the more unexpected one. Osteocalcin is the latest gene to appear during osteoblast differentiation, and you cannot find its expression in osteoblasts. before E16.5, and even at E16.5, it's rather low compared to what it is at E17 or 18.5. And of course, it's an osteoblast-specific gene. So E16.5 is when things starts in the brain. Yet, if we measure circulating osteocalcin in osteocalcin null embryo, we see that we can measure osteocalcin at E14.5, suggesting that maternal osteocalcin crosses the placenta and can be found in the serum of the mice or the embryo. And indeed, when we use this machine, which is called a placentometer, we could show that osteocalcin start to cross the placenta at E14.5, does it much more at E15.5, and obviously much more at E18.5. And so we wanted to measure the importance of maternal osteocalcin in the phenotypes I showed you. And to do so, we crossed father and mother that were osteocalcin knockout. So there is no osteocalcin in the system. And then we inject, once the female was plugged, we injected osteocalcin once a day in these mice. And then let the pregnancy develop and analyze the mice 3 months later. And what we observed is that in mice that received osteocalcin during pregnancy, serotonin content in the brain was normalized. Dopamine content, which was very low otherwise, was normalized, and the same thing for norepinephrine. The anatomy of the brain you can appreciate on this slide. I'm not going to move my mouse. Is reestablished to virtually normal anatomy. And memory measured by Maurice Watermans is not completely normal, but it's very close to be normal. And it's not completely normal because osteocalcin postnatally has also an influence on memory. And so that was really, to us, an eye-opening experiment because it showed that osteocalcin of the mother was controlling physiological function, and this applies only to memory. Anxiety and depression is controlled only by postnatal osteocalcin. But this experiment told us something else. It told us that if we inject osteocalcin once a day, and the half-life of osteocalcin is 15 minutes, the genetic program that is triggered by osteocalcin is so important that we normalize more or less everything. Okay, so all these experiments showed that osteocalcin is necessary to prevent anxiety and to increase memory. But it did not say anything about another fundamental characteristic of an hormone, which is that it has to be sufficient to fulfill this function. So you probably all know that 6 or 7 years ago, the group of Tony Weiss-Corey showed that young blood, when it is injected in old mice, can reverse the impairment in cognitive function. And I think it's fair to say that they have not necessarily found the molecule present in young blood that is responsible of this function. And it is a very difficult thing to do. We were helped here by having one candidate molecule which we tested. So here we injected blood of old mice in old wild-type mice, and we did elevated pulse mass test. We did the same thing for Morris water maze. And you can see that these mice are anxious and have a defect in cognition. I'm sorry. If, on the other hand, we inject the serum of young wild-type mice in old mice, we improve cognition, and it's significant. If we redo this experiment with a serum of young osteocalcin null mice, there is no improvement of memory, and if anything, it is worse than it is by injecting edge into edge. And as a control of this experiment, we took serum or plasma of young osteocalcin null mice and spiked it with osteocalcin, and we restored memory. It's important because it shows that this function is not a developmental function. It is a function of osteocalcin itself. And that led us through other experiments to clone the receptor for osteocalcin in the brain and to study its function, which I'm not going to show you here, and show that indeed this other GPCR called GPR 158 is necessary for cognition and to prevent anxiety in mice. So after all this work, what we had is something puzzling. We had an hormone made in bone, could have been made elsewhere, but it was made in bone, that is necessary for glucose homeostasis. I did not show it to you. That is necessary for adaptation to exercise, necessary for testosterone synthesis and male fertility, and necessary to go to medical school and not to be anxious. And the question we were facing is, what is the common thread between these functions? And a priori, I'm like you, there is none. Nothing in common between insulin synthesis and anxiety, or testosterone synthesis and adaptation to exercise. But there is a second level of analysis that look at the function, not at the molecules. And cognition was not invented to go to medical school. It was invented to remember where was the food yesterday, or where was the predator an hour ago. And even if you have, as an animal, the best memory, if you cannot run, it's going to be a very long day. If you cannot mobilize glucose, you will not be able to run. And during danger, there is an increase in testosterone synthesis. So under this light, and it is a stretch, I admit it, you could say that the function of osteocalcin may be needed to escape danger. And then the next question is to know, is this anecdotal, it is just specific to osteocalcin, or it is true for other function of bone? And this goes back to the initial question. Why did evolution invent bone in the first place? So you are sitting at your computer, and if the ceiling falls on your head, you will be in bad mood, but you will be alive because bone protects your brain. We have in our middle ear, 3 little bones without which we cannot hear. And being deaf and living in the Amazonian forest is a tall order. As I told you earlier, bone is here to repair fractures. That's for the structural aspect of bone. As for the ambulatory function of bone, without bone you cannot walk or run. So it seems that both classical and endocrine function of bone suggest that it is a tool to sense and escape danger. And if it is true, it can be tested because the quintessential function to escape danger is the acute stress response. So I'm going to give you several definitions of acute stress response, one with words and one with an image. It's an evolutionarily conserved physiological process that maintains or restores homeostasis in animals facing danger. It manifests itself by vasoconstriction, increased heart rate, increased respiration, and high energy expenditure. And there are other manifestations that cannot be studied easily in mice. And in fact, the way it manifests itself, you submit a manuscript to a journal, you think it's the best manuscript ever written. You wait, the reviews come back, and that's how you feel after reading the review. And that is exactly the best representation of the acute stress response. Molecularly, and we have to speak of it, of course, the sympathetic tone is the main, if not the only mediator of the acute stress response. What triggers, however, the increase in sympathetic activity remains unknown for now. If it is an endocrine mechanism, it can only be a peptide hormone because only peptide hormone would act within minutes, if not seconds, to trigger a reaction. By that I mean it cannot be a steroid hormone, it cannot be the glucocorticoids because they are steroids. And if bone is a survival tool, is it implicated in the acute stress response? So, what is a stress hormone? It has two criteria and only two criteria. Its circulating level should rise promptly during an acute stress response. And as I will show you, it should enhance physiological processes that are recruited during the acute stress response. And so we tested that for osteocalcin, not to look for another function of osteocalcin, but to look for an identity for osteocalcin as a hormone sensing and reacting to danger. And regardless of the type of stressor that we use, restraint, shock on the foot or the feet, or fox urine, there is a 2 to 5-fold increase in circulating osteocalcin within 2 minutes of the exposure to the stressor. And this increase in circulating osteocalcin precedes the increase of glucocorticoid during stress and remains stable for 3 hours. It's true in mice, it's true in rats, and it's true in humans that were asked to give a talk without warning. Through a chemogenetic analysis, we could show that this reaction requires the integrity of the basolateral amygdala, and if neurons of the basolateral amygdala are destroyed, and animals are subjected to stressors, there is no increase in circulating osteocalcin. The release of osteocalcin during an acute stress response is independent of the sympathetic tone. Taking advantage of the work we have done on the mediation of leptin regulation of bone mass, we know that the beta-2 adrenergic receptor is the only adrenergic receptor present in osteoblasts. And if you treat mice with beta blockers, you decrease the heart rate, which means that you did cause sympathetic inhibition. but circulating osteocalcin goes up, as it should if these mice are exposed to stressors. It is also independent of the adrenal glands. If you take adrenal-ectomized mice that have no corticosterone because you remove the gland, and you stress them, circulating osteocalcin goes up even more, if anything, than in wild-type mice. So at that stage, all we had is that stressors have to go in the brain, signal in the basolateral amygdala, and something has to happen between the brain and osteoblast so that osteocalcin is released. And we did not know what osteocalcin was doing, if it was doing anything during the acute stress response. So we address the first aspect of this slide, what is going between brain and bone. And I'm not going to go into all the details because some of them are purely important for bone biologists. Suffice to say, we wanted to know if the mediator of the stress or stressors was in the blood or was in neurons. And to do that, to distinguish between these two possibilities, we did a simple experiment. What we did was to put osteoblasts, wild-type osteoblasts from wild-type mice in culture. And we cultured them in the presence of osteocalcin, of serum coming from osteocalcin null mice. So all this osteocalcin that can be detected in the serum will come from the osteoblast. And we took serum from osteocalcin null mice coming from mice that were not subjected to stress or subjected to stress. And you can see that the same quantity of osteocalcin is present in the serum, in the extracellular medium, indicating that blood is not what mediates brain to bone. Instead, if we block neuronal signaling through chlorhexidine, we block the increase in osteocalcin circulating levels during the stress response. And so we tested many hormones to verify absolutely that there is no involvement of hormones during the stress response by treating osteoblasts with this hormone, and you can see that really none of them increase the release of osteocalcin in the medium. We also treated many neurotransmitters, and none of them had any effect on the release of osteocalcin until we tested the only one that we should have tested, which is glutamate. And glutamate increase the release of osteocalcin because glutamate is the business end of osteocalcin, as I told you earlier. So glutamate has many identities. It is, for most scientists, the most powerful activating neurotransmitters in the brain. It is also present in the peripheral nervous system, where its function is or are unknown. It enters cells through transporters, but it has another identity. It is an amino acid. And as an amino acid, it can be carboxylated in osteocalcin, and osteocalcin is inactive, or it can be uncarboxylated, and osteocalcin is active. So that's a schematic representation of that. Osteocalcin is a 46 long amino acid protein with 3 glutamate residues. And then there is an enzyme, gamma carboxylase, that when it carboxylates this glutamate residue, transforms them into GLA residues and makes the protein inactive. So briefly, what we showed is that there are neurites, that glutamatergic neurites in red, that about osteoblasts in white, and there are synapse-like structure between glutamatergic neurons and osteoblasts. And molecularly, we could show that if we block the release, the entry of glutamate in osteoblasts through a transporter called GLAST, we could block completely the release of osteocalcin during stress. This receptor, this transporter is GLAST, and briefly, because time is going on, GLAST is osteoblast specific. And if we knock out GLAST, then there is no change in circulating osteocalcin in the serum. We have not tested if GLAST mice have a stress response because these mice are very sick. But we have tested that glutamate is a competitive inhibitor of gamma carboxylase. And what we have is that during stress, glutamate is released from glutamatergic neurons, enter in osteoblasts through GLAST, act as a competitive inhibitor of gamma carboxylase, and as a result, uncarboxylated osteocalcin is released, and something happens. And this something is something we did not know. So again, I repeat, a stress hormone should regulate the physiological functions that are mobilized during an acute stress response. And we tested this assumption for osteocalcin. And we showed that there is no stress response in osteocalcin null mice, whether we look at energy expenditure, heart rate, or respiration. And the same is true in mice lacking the receptor of osteocalcin in peripheral tissue called GPRC6A. So osteocalcin does not inhibit or activate the sympathetic tone in the peripheral nervous system. But what it does is that it activates the parasympathetic tone. And in mice that lack osteocalcin signaling, heart rate variability, which is a marker of parasympathetic activity, is increased. Gastrin, whose secretion is under the control of parasympathetic tone, is increased. And the expression of CHAT, VCHT1 and CHT1, that are genes needed to make and reuptake acetylcholine, is increased in the absence of osteocalcin or its receptor. We could show that there are postganglionic parasympathetic neurons that express the receptor of osteocalcin in green and CHT1 and other marker of parasympathetic neurons. So in a nutshell, the autonomic nervous system is made of two arms that are opposing each other, the sympathetic activity, fight or flight response, and the parasympathetic activity, the couch potato response. We tested that osteocalcin affects one or the other by injecting osteocalcin in mice, and we can see, that there is no effect of osteocalcin on the sympathetic activity. But it decreases profoundly the sympathetic activity, leaving the sympathetic tone unabated, and the stress response then can now develop. It's also true in humans, although I don't have time to show you the data. So in a nutshell, stressors act in the brain, recruit glutamate, and that enters in osteoblasts through GLAST to release osteocalcin that inhibits the parasympathetic tone, and as a result, the acute stress response can develop. So this is another representation of what I just said. And I just want to tell you where we are now, because we are doing more experiments to explore the biology of osteocalcin as it is linked to survival. A genetic approach reveals how little we know about physiology. We know the molecular physiology, but we don't know the physiology. Bone is an endocrine organ that regulates an unexpected number of physiological functions. We did not expect that. And this occurs through conventional and maternal delivery of the hormone. Its endocrine function suggests that bone may belong to a broader biology of danger. And I will stop here. I hope I was not too long. But thank you again for inviting me.
Thank you very much, Professor Karsenty. That was really fantastic, and don't worry, you didn't go on too long. It was a really interesting talk. I would invite people to write their questions in the Ask a Question panel at the bottom. But I'll start off with a question that I had about this last section of the talk on the acute stress response. So I found it really interesting that glutamate seemed to act not to act on osteoblasts through glutamate receptors, but to be transported into the osteoblast and act within the cell. So do you have any idea of where those glutamatergic projections are coming from? Where are the cell bodies of those glutamate neurons? Or
yeah, so that's really what we want to find out, and it is an extremely important question of neuroscience and bone biology and biology of danger. We don't know yet where these glutamatergic neurons or axons are coming from, and we are looking for it. We are not neuroscientists, but we are trying to address this question. It is a fundamental question. You're absolutely right.
yeah, I mean, my impression is that overall, relatively little is known about peripheral glutamatergic neurons within the body.
Yeah, so what we would like to know is where is the known body of these neurons and where do they project in the brain? But no, we don't have the answer yet to give you. But this,
Yeah.
I realize it is a very important question. I'm fully aware of it.
So we have a question here from Zita Sanch. So she says, what an interesting, amazing talk. She wants to know, is it known whether osteocalcin is produced by specific bones? And also, is the innervation of bones generalized across the body, or is it specific to particular bones?
So we think among osteoblasts, there is heterogeneity, and there is a subpopulation of osteoblasts that is enriched in the ability to express and secrete osteocalcin. But this osteoblast, we have no reason to believe would be present in only one bone versus another. And the second part of the question, I'm sorry.
It's about the glutamatergic innervation. Is that also present in all bones?
As far as we can tell, yes, it is present in all bones, but we have not. There are 227 bones in the body. We have not looked at all of them, but at few of them, and they all have glutamatergic neurons. Yes.
Mm-hmm. Okay. Another question here from Carl Kubairu, who says, fascinating talk. You touched upon the evolutionary aspects of bone. So what is known about osteocalcin evolution, and what does it tell us about the evolution of bones as an endocrine organ?
Yeah, so osteocalcin is a bone-specific molecule. It appears during evolution with bone. There is no osteocalcin in animals that have no bone, just like there is no leptin in the animals that have no bone. And what it tells us is that bone was not invented only as a scaffold, but because of the energetic cost of bone modeling and remodeling, it was endowed with an ability to regulate physiology that is absent in invertebrates. And what it says, it whispers it, but it says it pretty clearly, is that physiology is regulated differently whether you have bones or whether you don't have bones. And this is because of the energetic cost of bone modeling / remodeling.
Mm-hmm. Yeah, absolutely. So another question here from Yag, who asks, who says, a very interesting talk, thanks. Does exercise stimulate osteocalcin production?
This is a very insightful question. Yes, exercise increases the secretion of osteocalcin 60% in humans, 400% in mice. It's a main regulator of osteocalcin, and exercise is a proxy of escaping danger.
Mm-hmm. So one more question here. It's fascinating talk. Why do you think Manolagas obtained contradictory results? I don't know about what they're referring to, but I'm sure you're aware.
Yeah, so that's a fascinating question because Manolagas did not obtain different results. Manolagas commented on the work of others. And I can go through the pitfalls, technical pitfalls of the work of others. They look at muscle mass, not muscle function. They did not look at cognition or acute stress response, as far as I am aware of. But I won't do that because I'm not here to backstab people that are not present. I would just say that one has to look at the literature in total, and there are approximately 150 papers or 250 papers showing that osteocalcin is an hormone in mice, monkeys, and humans. And I would also say that probably you were not around, and it is a privilege that you were not around, in 2000 when people were publishing papers showing that leptin has no role in appetite. And for them, like for others, time is a great master.
Mm-hmm. Mm-hmm. Excellent. Well, thank you very much for your answers to these questions. I think that's all the questions we have for now. Are you still available for the
Yeah,
for a meet the speaker session afterwards? So I sent you the link via email. If anyone, I know some of you signed up already, and I've sent you the link. If anyone who didn't sign up initially would like to join our meet the speaker session, we still have a few slots available, and I can send you the Zoom link. We'll meet there in about 5 minutes or so. Otherwise, I'd like to thank you very much, Professor Karsenty, for joining us today. It was a really fascinating talk, and I think you really did a great job of fitting your work into the kind of interests of our audience, and I'm sure it's been really interesting for them.
Thank you.
And for everyone in the audience, we'll be back in January. And keep an eye on our email list or on our Twitter account to see for more information about upcoming talks, as well as on our website, brainbodyinteractions.org. Look forward to seeing you again, and thanks very much.
So Sam, I wait for your email?
Yeah, I just sent you the email.
Okay, thank you.
Thank you.